any hard feelings over the strobe light incident? Some people don't know what I'm talking about there. Yeah. So my presentation is going to be about antioxidants in general and beta-carotene specifically. And we'll talk a little bit at the beginning about what antioxidants actually are as molecules, and we'll transition into sort of a history of the research surrounding beta-carotene research, and then some more modern scholarship, and then we'll end with what the local research group at Taylor University ended up doing on this topic. So any sort of talk about antioxidants really should probably start with a discussion of reactive oxidative species. And if you attended Dr. Hammond's talk last year on this, oxygen friend or foe, this should pretty much be a recap for you, so sorry about that. But I guess the long and short of it is that most oxygen, or most likely oxygen to live, the drawback to a reliance on that element is that oxygen is extremely reactive and unfortunately sometimes reacts in ways in biological systems that are maybe not conducive to life or where they're actively damaging. When that happens, that's called oxidative stress. All right. So reactive oxidative species are actually the compounds that are causing this type of damage. And a hallmark, I guess, a reactive oxidative species is a species or the compound that contains oxygen, and it's typically a free radical. There is, I guess, some exceptions, or one exception, like singlet oxygen is an exception, but in the vast majority of cases, it is a free radical. All right. So there's many different molecules. I already mentioned singlet oxygen. A big one is superoxide ions. Here's the mechanism for how those form. And actually, these are an reactive oxidative species, but they also they're also the precursor to other, maybe even more damaging reactive oxidative species. And then we got hydroxyl radicals here is another sort of example of that. That's just a hydroxide ion that's missing an electron. So some examples of oxidative stress, or what are some ways specifically that we occur by oxidative stress? Well, one example, or a big example, is DNA oxidation. And in a very specific instance, this can actually be seen as a good thing. Epigenetically, there's at least, I came across at least one research paper that seemed to indicate that a gene could be switched off using DNA oxidation. But in the vast majority of cases, it's not. It can cause mutations. In extreme cases, it can even compromise the structure of the entire DNA molecule. I believe it attacks at the hydrogen bonds that hold the two strands together, so it can cause denaturation that way. Reactive oxidative species can cause damage to protein side chains, or proteins as their amino acid side chains. If you know anything about tertiary structure of proteins, you know that those side chain interactions are very important to tertiary structure. You think about like disulfide bonds and cysteine molecules. So obviously oxidation there, if those can be compromised, that can compromise potentially the whole structure of the protein, and protein structures everything. The function follows the structure. In enzymes specifically, reactive oxidative species can oxidize coenzyme molecules, and that can have the effect of deactivating them entirely or making them less active maybe. And probably most relevantly for our discussion here, reactive oxidative species can damage lipids via lipid peroxidation. I've got the mechanism up here. It's probably what's most important to realize that in the absence of any sort of antioxidant molecule or any way to halt the propagation step of lipid peroxidation, you could get a cell membrane which is made of phospholipids. It could be compromised, and you could have holes there or fluidity problems with it. So what are antioxidant molecules? So broadly, antioxidants are any molecules that combat oxidative stress in biological systems, and I guess there's really two ways you could do that. The first way is you could, maybe try to stop the formation of reactive oxidative species or free radicals, before they even form. The second one is probably more relevant to our discussion here, because this is sort of a category that beta-carotene falls into, is that you could try to neutralize free radicals after they form. There's two additional broad categories which we're thinking about it. There are hydrophilic antioxidants and lipophilic. Beta-carotene is lipophilic, so it resides mostly in stopping lipid peroxidation, which we talked about in that last slide. All right. So several examples of antioxidants, I mean dozens of examples. If you Google antioxidant, you look on the Wikipedia page, there's probably 24 examples they just have listed right out there. So several enzymes fall under this category. Superoxide dismutase is probably the one that comes up most often. Vitamin E and beta-carotene, which we'll be talking about pretty much for the rest of the day. So beta-carotene has an interesting research history. And actually a lot of antioxidants sort of follow this general pattern where they were discovered a long time ago, were first isolated a long time ago, think on this time scale like 100 years ago. And it's only relatively recently, think the last 40 years, where their antioxidant properties have really been explored in any amount of depth. So beta-carotene is certainly not an exception to this rule. It was first isolated in 1831, so I mean that's close to 100 or 200 years ago. They were trying to isolate plant pigments. So that's actually one way that beta-carotene is used today industrially, is it actually is used as a food dye. It's extremely red. I can tell you from working with the concentrated thing. Its empirical formula was discovered in 1917, close to 100 years after it was originally isolated. And 2 years later, its relationship to vitamin A was determined by Steenbach. And this one is particular. This location on the research timeline is particularly important to me, or special to me, because Steenbach was a researcher at the University of Wisconsin-Madison, where I will be attending medical school. The STEM library is actually named after him. And another interesting thing about him is that this sort of reaction is used to fortify milk, or was used to fortify milk with vitamin A. And it's sort of the process that he came up with. He took a patent out on this process as soon as he figured it out, and it's one of my primary sources in my paper. It cost him $300 to file for the patent, and a couple years later he was a multimillionaire from it. So good investment on his part. And then its structure was discovered in 1930 by Paul Kerr, for which he won the Nobel Prize in chemistry in 1937. And you might be wondering, well, that's kind of weird. Like, obviously we don't give Nobel Prizes out every single time we figure out the structure of a biomolecule. And that's true, except for, I guess what was special about beta-carotene was really the first time that a vitamin or vitamin derivatives structure had been discovered. Okay. So let's talk about there's a little bit more modern research. I mean, at least it's not close to 100 years ago. In the 1970s, I guess we knew about oxidative stress for a little bit, but in the 1970s, the dangers associated with it became particularly well known. Actually, what happened was there were one major study and a couple peripheral studies that seemed to indicate that higher levels of free radicals in people who had higher levels of free radicals seemed to be at an increased risk for cancer. So in the early 1980s, there were several papers published. Again, there was really one major paper and a couple of papers sort of confirming this, that there was maybe a link between people who consumed high amounts of beta-carotene in their diet and lower rates of cancer. I'm just going to stress this now because it becomes important later. You have people who were consuming high amounts of beta-carotene in the food that they were consuming, not necessarily people who were taking beta-carotene supplements. All right? But some people in the scientific community wanted to explore the cancer prevention or cancer preventative properties of beta-carotene as like a supplement, so they moved to animal trials with that, particularly rats. And so it was shown, several times that rats in an experimental group that were exposed to beta-carotene, and obviously not food, so they were taking, what would be akin to a beta-carotene supplement, or they were being injected with it. Rats in those sort of experimental groups experienced less tumor growth relatively than rats in a control group. So it looked promising for that idea until they started human trials in 1919, or 1992. And it was shown, well, the first study that was done, and you have to realize that obviously they weren't testing people like rats, like you took humans and exposed one group to beta-carotene supplements and the other group wasn't. Beta-carotene had actually already been on the market as a supplement even for a while, I believe since '56. And I guess if that sounds surprising to you, maybe you shouldn't, even though they hadn't maybe explored the health benefits of beta-carotene a lot, supplements really don't need, I can tell you right now from working at a pharmacy that the supplements are not rigorously tested.
No.
We sell plenty of what I would call snake oil supplements, and beta-carotene may be at this point of history fall into that category. But suffice to say, there was a population of people who had been taking beta-carotene supplements at this time, and so we were able to compare their health outcomes, in the 1990s with people, a control group of people who hadn't been taking beta-carotene as a supplement. And the first such study sort of seemed to indicate that, yes, there was a modest, decrease in cancer rates in people who've been, supplementing with beta-carotene. But later follow-up studies, and I mean almost immediately, so pretty much within the year, there were several studies that could not replicate that finding. And later in the 1990s, actually mid-1990s, there were more than one study that seemed to indicate an increased risk of cancer associated with beta-carotene supplementation. So suffice to say, the scientific consensus at the end of the 1990s was that beta-carotene was probably not going to be a good bet for supplementation, at least if you're hoping to get cancer prevention properties out of it. So where does that leave modern research regarding beta-carotene? Well, if you think back to the original study that was done, that was demonstrating a link between foods high in beta-carotene and in their diet and cancer prevention or decreased cancer risk, you should realize that maybe even if beta-carotene maybe by itself in this sort of concentrated form doesn't prevent cancer or give you sort of protection in that way, maybe it's beta-carotene with all the other chemicals found in spinach, which is a food high in beta-carotene. Maybe that's what causes it. So the modern research has really moved towards just quantifying the amount of beta-carotene in food, and some of the research has moved towards trying to figure out what ways of processing our food can we make beta-carotene more bioavailable or actually get that. So the research at Taylor University has moved in that direction more. Can we come up with a way to quantify the amount of beta-carotene in papaya is specifically what we were looking at, and is there a way in which maybe we could treat papaya that could make the beta-carotene more bioavailable? All right. So our research was a little bit different than what I've noticed other research in this area tries to do. A lot of times what people do when they're trying to quantify beta-carotene is they put it through HPLC. They use HPLC to figure it out or to quantify. And that's not what we did. What we did is we took DPPH, which is a free radical, and is obviously going to react with beta-carotene. And in its unreacted state, it absorbs very strongly at 450 nanometers. And in its unreacted state, it is clear. So what we did is we took papaya extracts and we reacted it with DPPH stock solution that me and David prepared. And we were able to monitor the reaction by using a spectrophotometer to watch as the DPPH disappeared. So you could tell, obviously, less DPPH, at the end of the reaction is going to indicate relatively more antioxidant property in a given extract. So that's sort of what we're going to do. I'll first walk us through the extraction procedure. It's not horribly complicated. I think really the thing to take away from this is that for each papaya that we started with, or type of papaya we started with 30 grams of it, and at the end of the extraction procedure, we ended with about 100 milligrams, or 0.1 grams. And that was pretty consistent, and usually what we ended up with was a solid or sludge. Only once did we get a really good solid. The rest was more of a sludge that it wasn't evaporating in the road evapor. It was not going anywhere. So anyway, I guess what I'm emphasizing that we got the same amount at across papayas is that any sort of difference in antioxidant property that we would observe in the extracts, I think you can probably safely say came from differences in how much beta-carotene was bioavailable or for reaction in the original papaya. All right. So we actually used two different ways to use the spectrophotometer in two different ways to explore this. So the first one, what we called, is called the simple scan procedure, and essentially what we did is we took some papaya extract and we mixed it with DPPH solution, and we took a quick scan at the beginning, and we waited 90 seconds, and we took a quick scan after that, and you could, difference in absorbance there would tell us relatively how much DPPH had vanished, since the reaction, and obviously more DPPH gone, right, or more DPPH reacted would indicate relatively greater antioxidant property in the extract, all right? So here are our results for that, and one thing that you should notice is that we did it with several different volumes of papaya extract, all right? So if you just follow one down, we'll look at fresh papaya, I think that's a good example. You'll notice that, at 40 microliters of extract solution, that had relatively more DPPH remaining and therefore less antioxidant property than 50 microliters of papaya extract. That's a good thing. It's a good thing we see this downward trend for every single one because something would be wrong if we didn't. I mean, so I think probably this is the best one to look at, this circular group right here. But you can see that frozen papaya, all right, had the least amount of DPPH remaining after 90 seconds, which would indicate the most amount of antioxidant property. And it's important to realize that the frozen and fresh papaya came from the same papaya. It was literally a papaya me and David cut up. The frozen papaya got tossed in the freezer for like a week or two, and the fresh papaya we did pretty much right then. We extracted pretty much right then and there. So it's not like we were working with different papayas and maybe one was more ripe or something. This was the same papaya, and there was a significantly different, amount of antioxidant property demonstrated between those two groups, which was interesting. We'll talk about it in a little bit in a second. These two, I think, are maybe too close to call between the boiled and the canned, based on this procedure. On which one is closer, or demonstrates more antioxidant property. You can see that the trend kind of switches a little bit. It's kind of hard to tell here. But luckily, we were able to resolve the differences there, I believe, in our next procedure. So we also did, we used a kinetic scan to try to figure out or quantify the amount of antioxidant property in papaya extract. So what we did here is very similar. We mixed papaya extract with DPPH solution and put it into the spectrophotometer, but instead we calculated a reaction rate, and it makes sense, if the reaction rate is higher, or relatively higher, that would indicate relatively more antioxidant property for that extract, right? So in theory, these are the results for this, and I'll just real quick point out some things. There's a slide that sort of puts the results of the two different procedures side by side. But you can see fresh or frozen papaya leads the day again, with fresh papaya following closely behind. And actually, one thing that was interesting here is you can see that the difference between boiled papaya and canned papaya will resolve much better. All right. So here's the sort of all put together in one easy way to look at it. You can see that frozen papaya and fresh papaya extract both have there's a lot of correlation here, I guess, or they feed into each other well, the results of the different procedures. And I'm hesitant to call this necessarily a contradiction. I just don't think that the simple scan procedure was very good at resolving the difference there. I would have to say, after looking at our kinetic scan results, that the boiled papaya probably does demonstrate a little bit more antioxidant property. So what would this mean for sort of the field overall? It's interesting that the frozen papaya, and I mentioned this earlier, had more antioxidant property than the fresh. What would cause that? There's a couple different theories, and probably the one that is the most relevant and probably that we're hoping for is the case. It's possible that the process of freezing and thawing lysed some cells, or opened those up, and made the beta-carotene more bioavailable than than it would if it was fresh. And whether or not this is significant to the field, or whether or not, making beta-carotene more bioavailable in this way actually has a real-world application for preventing cancer, I guess that's sort of debatable, and we need more research in that area. But what's interesting with our results is that we did show that I think that freezing, at least in some capacity, probably does make the beta-carotene more bioavailable. Yeah, which is a goal of my research. So that's it. Any questions?
So if your theory on why the freezing worked, do you think that that could be matched maybe if you were to blend papaya, and then similarly, like why wouldn't cooking, like why does cooking produce kind of the opposite effect? Does cooking have any effect on like cells?
Yeah. So probably I would imagine that denatures papaya or beta-carotene, or could make it. I mean, if you've seen the beta-carotene concentrated, we store it in the fridge. We don't want to store it at room temperature; it degrades. And actually, Steenbach, that guy who figured out that vitamin A comes from beta-carotene, you can make vitamin A from beta-carotene. All it takes is ultraviolet lights to really convert it. It's not a horribly stable molecule, as my understanding is. So I'd probably say that's probably why.
But what about like if you tried to blend
it and lyse the cells that way, you mean? What we did actually, if you go back to the extraction procedure, homogenizing is essentially blending.
Oh, okay.
So we did do that. Now, as for I don't know if there's a way to get an extract without blending it. David left. He came up with the extraction procedure. So. Great question. Great question. Yes, Dr. Sichula.
So the beta-carotene. What is the structure? Do you go back to the structure again?
Yeah. Right here.
Yeah. So this compound you are saying when fresh and then when dry, the antioxidant properties were varying.
Yes.
So the question to you is that this molecule, is it unstable at different temperatures, or does it decompose? Have you investigated?
I have not. I have to say we did not investigate that. We were mostly we didn't actually work. The only real time we worked with beta-carotene concentrated, which we did do. If you go back to our let's see. What was the question again, Dr. Sichula?
Yeah, so you are saying, when fresh, okay, for example, a fresh papaya and then dried, the antioxidant properties were varying. That's
Yep.
according to your findings. So my question is, because the antioxidant property is supposed to be due to the presence, the amount of the beta-carotene, isn't it? In the papaya, isn't it? That's supposed to be, because that's one that produces antioxidant properties. So the question is, why is that the variation in the properties? Is that due to the fact that the compound is unstable at high temperatures? High temperature, or why?
Yeah,
because that doesn't make sense when fresh. Because we are dealing with a compound.
Yeah, I don't think that for the fresh versus frozen, I wouldn't say that. I mean, obviously, I don't think that beta-carotene just appeared in the frozen.
Right.
I would say that somehow in our so if you think back to our extraction procedures, it was obviously we froze it before we even started extracting it. I would probably say that in the extraction procedures we strained off in the fresh. We strained off there was a solid component. And I think the whole idea behind the freezing thawing hypothesis and lysing the cells is that when you strained off the solid, there were probably some intact cells that still had their beta-carotene sitting in there.
Okay.
Whereas in the frozen instance, those were all relatively more lysed.
Okay, that makes sense. Then one question you had whereby you did the extraction and then you where you said the liquid was placed in the wrap into a solid or a sludge was obtained. So the question is the difference in a solid and a sludge. So what's the reason of getting that? Is that when you're doing the extraction, are there other compounds that are present in the papaya with the chemical as well?
No, that's a great question, and I would have to say that absolutely yes. There are other lipophilic biomolecules in the compound. One thing though that I think is important to mention is that I see what you're saying. I don't know if it's horribly relevant to what we were trying to uncover, because even if there were other molecules present or lipophilic molecules present, I mean, we still demonstrated that it could reduce the DBPH. So I guess.
My question is that maybe you were not getting the solid, maybe the presence of impurities sometimes makes the compound not to be solid. When you're doing extraction, when you evaporate your compound, I think most of the students who do research, they will attest to that. When you evaporate the solid, your compound, instead of getting a solid, you get a liquid due to the presence of impurities. So what I'm trying to say is that those compounds, maybe there might be other compounds that are present in the papaya that causing to that.
You could be right. Yes, Dr. Kuka?
When you do the extraction, and you do it at room temperature?
Yes.
And like you were saying, that if you were sure that the antioxidant is from the beta-carotene, there's no other compound in the papaya that's causing.
I can tell you that I'm not sure 100%, but here's something that would seem to indicate that it was. I've smelled beta-carotene because I've smelled. It's actually quite pleasant. It is not a bad thing to smell. And the extract, I can tell you that the extracts sort of smell like beta-carotene. And I know that's obviously, that's not like a rigorous analytical technique, but the color was there and the smell was there. So I got sight and my chemical sense then. Yes, Dr. Kroll.
Yeah, I guess all of us who've done organic extractions have got the same kind of questions. Because I have the same kind of thoughts about, well, what else is in there? Yeah, and I know beta-carotene isn't terribly stable because back when I was doing organic, we had a lab that we were doing plant pigments, and I looked into getting some beta-carotene as a reference compound and found out A, it's really expensive to get it pure, and B, it decomposes quickly. So, I mean, that's an issue that you have here. One question I had was, did you do multiple trials of these things? I mean, is it one category of each, or, I mean, how much consistency was there between like a trial with frozen papaya, did you do it like 5 times and you got the same kind of results?
10 times.
10 times, okay, that's good. As long as the variance wasn't too great within that 10 times of the one kind.
As you were describing beta-carotene, it just occurred to me how it's like the perfect snake oil. Like, it's expensive and it decomposes quickly,
Yeah,
so
yeah.
eat it quickly.
Maybe because it was one of the reasons.
Dr. Hammond?
Perhaps one of the things that you could be getting at is bioavailability,
Yeah.
the beta-carotene or other antioxidants. But I'm not sure that it would show that because plant cells have cell walls that are hard to break open. So when you digest them, you can break them open and extract, these chemicals from them. But in the extraction technique you're using, I'm not sure that that's happening. So the freeze-thaw thing that you were talking about is probably true. And that explains more as to why you have higher beta-carotene
In the
in your
frozen.
extract than perhaps anything else.
I agree. And I did hear one theory. I ended up having to explain my research, or my interviewer at the University of Wisconsin-Madison was actually extremely interested in this. And she seemed to think that an alternative theory for what might be causing, the cell wall to break down or to increase the bioavailability might have been that some enzymes were being activated by cold. I did not think that sounded very likely.
Yeah.
So I guess, have you ever heard of that, Dr. Hammond? Enzymes being
Well,
activated by cold?
temperature is a possibility, but I don't see it in this case.
Yeah.
There are a number of enzymes that are activated by being heated. That's part of what goes on in say, cooking of meat at a certain point, but not by chilling, usually less active.
Yeah.
Oh, that's one. So when you froze it, freeze it, and what temperatures?
At what temperature?
Yeah. Do you leave it nitrogen freezing, or you just put it in the freezer?
We just put it in the freezer. It was there for 2 weeks. It was in the freezer in the stock room. Might still be there. I don't know.
I'm just thinking that probably when you do the process, the extraction, you probably could do that. You just test it, do a freeze and thaw like a couple times, and then maybe make a decision whether the freeze and thaw is responsible for the biopharmaceutical.
Yeah, because if you froze and thawed several times, you should see potentially a lot more beta-carotene. But that's a good idea.
So did any one of them taste better than the other?
Dr. Cameron, come on, we never eat afterwards. We only eat fresh.
The last question now, you are performing a reaction between, there's a compound, what compound is that?
DPPH. Dave, you want to take this one? You have your hand raised.
Diphenylpycryl hydrate.
Okay, so what are the purpose of doing the reaction? Could you go back where you described the reaction? I just didn't get it.
That's a standard reaction for this.
Yeah, what's
So
so
what
great
you are
about
trying to do when you do that reaction? Yeah. What are the purpose?
what's the purpose of using
Yeah.
DPPH? Okay, so DPPH absorbs, what's convenient about it is that in its unreactive form, it absorbs so strongly at 450 nanometers. I can tell you it's real strong. I mean, we were probably using, a milligram of this stuff, and it turned it pretty much completely black. And so as the reaction progresses, it becomes less black, it turns clear. So the reason we were using DPPH is because A, it's a free radical, and B, so it will react with any sort of antioxidant.
Okay, that makes sense.
It's a standard reaction for this kind of thing.
Thank you.
All right, thank you.